Hello, everybody. Nice to have you all here, ladies and gentlemen, and we welcome you to this webinar organized by the International Academy of Human Reproduction. And I see that we have a lot of participants from all over the world. So good morning to those who are following us from the Americas. For some of you, it's very early in the morning, so thank you for waking up that early for us. And good afternoon to those who follow from Europe and the Middle East, as well as good evening to our Asian participants. We are here today to listen to some updates on the latest steps on the way to a vaccine for COVID. And Professor Joseph Schenker will be our moderator for this webinar. Professor Joseph Schenker is the president of the International Academy of Human Reproduction. He is professor of obstetrics and gynecology at the Hebrew University of Jerusalem, chairman of the Department of Obstetrics and Gynecology at the Hadassah University Hospital, Jerusalem. His research was involved in experimental and clinical studies in the endocrinology of human reproduction, development of methods of contraception, and development of technologies in practice of assisted reproduction. He's interested in ethical aspects of reproduction and in gynecological and obstetric practice, and has chaired the ethical committee of IFFS and FIGO committee for the study of ethical aspects on human reproduction. He has produced publications in medical journals and several books in the field of obstetrics. So thank you, Professor Schenker, for being with us, and I leave to you the chairing of this session.
So thank you very much for the warm introduction. I am pleased to welcome you to the first webinar of the International Academy of Human Reproduction. This is the first webinar in our academy, which will bring interesting insights for our fellows and for a larger audience who are following our activities. The host our institution is the Academy of Human Reproduction, which was established in 1974, and it is a professional voice for researchers in the area and from clinical medicine, biological sciences, medical education, and population science. The Academy holds its congresses every 3 years, publishes a series of books on reproductive medicine for clinical practice, and promotes exchanges among scientists around the world. Our fellows are selected based on singular and significant contribution to the field, and they come from 5 continents. As was mentioned in our introduction and publication, the present subject which will be discussed by our scientists is the development of vaccine and monoclonal antibodies for the prevention and treatment of COVID-19. In today's webinar, we are able to direct spotlight at one of the group working interested in this research, the goal of COVID-19 vaccine, the Israel Institute for Biological Research. The Israel Institute for Biological Research is a governmental, cutting-edge, applicable research institute specialized in the fields of biology, chemistry, and environment science. The capabilities are scientific achievements and international reputation of these national institutes are the results of decades of research and development aimed at defense from chemical and biological threats. Can I have the next slide? I am pleased to introduce our speakers. The first one is Dr. Nir Paran. Dr. Paran is tenure researcher and head of Virology Section, Department of Infectious Diseases, Israel Institute for Biological Research. As you can see on the slide, his education was in Hebrew University, in Weizmann Institute, and his PhD was too in the Weizmann Institute. He at present is, as mentioned, from 2017, head of the Virology Section, Israel Institute of Biological Research, Ness Ziona, and his achievement is very well seen in his excellence awards which he received during his academic life, and especially I can say to receive repeatedly the prizes of Weizmann Institute is something special in our region. The next one. The second speaker will be Dr. Tomer Israeli, tenure senior researcher in Virology Research Group as the Department of Infectious Disease. Since 2015, he is a virology group of the Institute. His education, especially the interest for our fellows, is his thesis on angiogenesis in ectopic ovarian transplantation. Next slide, please. You can see in the list the same, his excellence contribution to the science, the awards what he gets through the years. So after introduction, I'd like to give the podium to the speakers, and it is mentioned that the public can participate and ask questions using QA button. This will be discussed at the end of presentation. Please.
Hello, everyone. So I will start. My name is Nir Paran, and together with Tomer Israeli, we will present our work on COVID-19. So this is the disclaimer, and we will focus our talk on the development of a vaccine and therapeutic antibodies. Short introduction to COVID-19 and SARS CoV-2. So coronaviruses are a large family of viruses. They cause disease in human and other animals, ranging from a mild common cold to severe disease and death. They are zoonotic and they are transmitted between animals and humans. And the disease manifestations are variable from respiratory symptoms to fever, cough, and up to severe cases with pneumonia, ARDS, and multi-organ failure and death. To date, there are no approved vaccines or antibodies for the treatment of or prevention of COVID-19, and this is the main topic of our work and presentation today. So a short introduction on the family. So this is a schematic representation of a coronavirus with the RNA inside the particle and the spikes that cover the viral particle and give its name like a crown. So there are coronaviruses that infect rodents or bats mainly, and these are mutated to infect other animals like camels, palm civets, and other animals. And from these animals, in certain cases, there's another mutation that results in a zoonotic infection of humans. So this list of 8 coronaviruses infect humans. They are part of them are from the alpha coronaviruses like the NL63 and the 229E, and most of them are from the beta coronaviruses, including SARS CoV-2, SARS CoV-1, and MERS that are circulating in the SARS CoV-2 in all the world and MERS in the Mediterranean. All the 3 coronaviruses that are have a red rectangle are cause severe disease in humans. We tried to see what is the relevance of coronaviruses SARS-CoV-1 and 2 and their association with the reproductive tissues, and we didn't find much. Maybe you will have more data to share with us, but there is no direct connection of the virus with the reproductive tissues, and no and the expression of the viral receptor and the proteases that are required for entry in the reproductive tissues is quite low. So it's mainly a disease of the respiratory system, and but in severe cases, of course, there is a disease in a multi-organ disease. So we are focusing on both a vaccine for the prevention or protection of the population from from being infected, and antibodies to treat infected patients, and maybe also to protect at-risk individuals. So to our vaccine, so we decided to develop a vaccine for COVID-19 based on the vesicular stomatitis virus. And the idea is that vesicular stomatitis virus is a virus that does not infect regularly humans. It's a virus that infects horses, it infects pigs, and some rodents. And it's a very simple virus genetically. It has one envelope protein named G protein that is marked here in green, and you can see that it covers the viral envelope. So this is wild type vesicular stomatitis virus, and we chose to replace the G envelope protein of the VSV with the spike protein of corona to generate a VSV delta G spike virus. And as you can see here in the schematic representation of the genome, and here a schematic representation of the virus carries the spike gene of SARS CoV-2 in its genome instead of the G, and express the spike proteins on the surface of the virus. And this is transmission electron micrograph of this vaccine. You can see the bullet shape of the VSV with the spikes all over its surface. They are decorated with antibodies against the spike, and they are gold labeled. So you can see the gold labeled nanoparticles decorating the virus, indicating that the virus is all over decorated with spikes. So this is the vaccine. And the benefits of this type of vaccine, it's for pandemic issues, for pandemic idea, is it's a very rapidly replicating virus. So it enables us to have to manufacture the vaccine in a scalable manner in cultured cells, in viral cells that are approved for vaccine production. The vaccine platform itself is safe, and the same platform with the envelope protein of Ebola virus was prepared by Merck, and over 300,000 people were already vaccinated with this FDA-approved vaccine for Ebola in Africa. It's approved by FDA and by the European authority. So it's a very safe and approved platform, and it efficiently induces immunity, both cellular and humoral immune response, following even single immunization, which is very important in a case of a pandemic. And there is low seroprevalence in the human population, especially in Israel, allowing to vaccinate the people without having to deal with prior immunity to the vector. So this is the system, and we have some publication that is currently under review, and we also have a patent that is was subjected about this vaccine. And a few words about the vaccine, the vaccine platform. So most of the data is from the Ebola vaccine. As I said, it's a very similar vaccine. And concerning issues of pregnancy and birth defects, which is very important in live attenuated vaccines. So based on the data so far, there are no data concerning major problems during lactation, gestation, and also during pregnancy, suggesting that the vaccine should be safe also for pregnant women. But we first think that we will start our clinical trials in people that are not pregnant. So about the vaccine, as I said, the vaccine has two modes of action. One, it acts as an antigen. The surface spikes all over the particle are recognized by immune cells as an antigen, and they generate immune response, both humoral and cellular. The other way is the virus carries the spike gene, and upon infection of cells following vaccination, the spike gene is expressed together with other viral proteins, and this spike protein is expressed on the surface, is presented on the surface of immune cells, resulting in induction of immune response, a very robust and authentic immune response, which altogether can have a protective ability even following a single vaccination. And here is a demonstration of the response following infection of cells with the vaccine in the middle panel. So you can see cells that were infected with the vaccine, and the antibodies recognize the spike protein. So the spike protein is expressed in vaccine-infected cells, similarly to cells that were infected with the SARS CoV-2 virus. We also wanted to make sure that the spikes on the surface of the particle are authentic in their structure and similar to the structure on the corona surface. So the antibodies, that the immune response that is generated by this vaccine will be able to protect against SARS CoV-2. So we compared the neutralization activity of human antibodies from convalescent people, how they neutralize the vaccine compared to their ability to neutralize the virus. And as you can see here, there's a very strong direct correlation between the two tests, indicating that the spikes on the surface of the vaccine are similarly structured like the SARS CoV-2. So altogether, we believe that this vaccine would be very potent, and Tomer will follow with the preclinical studies to support this.
was a base for first in human clinical trials. Safety and efficacy were examined in various animal models, as you can see here. We started with preliminary initial data from mice. These are special mice, which on the surface of their cells, there is the human ACE2 receptor, and that make them susceptible for the virus compared to wild-type mice. So these are a model that we just started, and I won't elaborate on it much. We also worked and established a model on hamsters, the Golden Syrian hamsters. This is our main model, and I will elaborate on this model shortly. And we also examined the rabbit and the pig model, and the examinations are mainly are regarding the safety of the vaccine. This is what you need in the preclinical stages, but we will show you also efficacy data on the vaccine, and the efficacy will be divided into the immune response and the ability of the vaccine to protect, especially the hamsters, challenged by the virus, the coronavirus itself. So as I said, the main model is the Golden Syrian hamsters, and we first established a model of the disease, and then we evaluated the vaccine, both regarding the safety and the efficacy in the Syrian hamsters. So first, regarding the model of the disease, as you can see here, this is a graph representing the weight change of the hamsters. The y-axis is the percent of the weight compared to the time zero of the challenge of these hamsters with the SARS CoV-2. And as you can see here, there is a nice dose response as the dose is higher. As you can see here in green, the weight loss of the hamsters is higher, is bigger. All of these studies, of course, are being conducted in a BSL-3 environment that we have in our institute. So we have a model based mainly on this loss of weight following the challenge of these hamsters. We also have a very new way of examining the effect of the virus on the hamsters. And we are using here a monitoring system which monitors the night activity of the hamsters. Here you can see mice, but it works also nicely, very nicely in hamsters. The idea of the system is that there is a beam, a laser beam, crossing the cage of the animals, and each time an animal crosses this laser, it is counted, and when the animals are feeling fine, there is multiple crosses during the night, and when the animals are sick, the number of the crosses are reduced. And as you can see here in the graph, in the blue, there are mock hamsters that were not challenged with the virus, and in the green, purple, and orange graphs, you can see the effect of the virus by the reduced activity. This is another way to show that the virus is affecting the hamsters, and there is a marginal disease. And of course, we examine the lungs of these animals. The virus is given intranasally. So we examine the lungs These are lungs 5 days post-exposure. So on the left side, you can see a naive, mock infected hamsters compared to on the right side, a hamster that was challenged with the virus, and you can see nicely the major differences in the H&E staining, and hopefully also in the viral stain in green in the lungs of this hamster. So after establishing the model with the virus, we started to examine the safety and the efficacy of our vaccine. The rationale of using this hamster is, of course, the disease that is caused by the virus. It is important to know to say that there are no other animal model, established animal model, that the virus is causing such a disease. This is the best animal model that is found, that is established until now. So this is a good model to examine treatment as a vaccine or other way of treatment. So regarding the safety, I will show you that data on the effect of the vaccine itself regarding different data, and the efficacy, as I mentioned, mainly regarding the binding antibodies that before the challenge, and the efficacy of the vaccine to protect the hamsters after the challenge with the SARS CoV-2. So first, regarding the safety, this is a regime of prime and boost. 14 days after the prime vaccination, we gave a boost. This is due to the regulation standards that are that we have. So we examined also the regime of the boost, and this is a graph showing the weight loss after the vaccination. And as you can see here, there is no any weight loss compared to the naive hamsters at different doses of the vaccine, from low dose to a very high dose. There is nothing that is happening to the hamsters regarding the body weight, and also regarding any local reaction that were absent in all of the vaccinated hamsters. Regarding the efficacy, this is a time schedule of the experiment. I will go over it fast. There is a single vaccination in this case in different doses. The vaccination is given intramuscularly. The hamsters, blood samples is taken from these hamsters 3 weeks after the vaccination in order to evaluate the neutralizing antibodies. Around 4 days after these hamsters are challenged with the SARS CoV-2, there is tracking of the morbidity. Some of these hamsters are euthanized in order to examine their lungs in histology and the viral load. And there is a tracking regarding the morbidity for about 2 weeks after the challenge. So regarding the first phase of the efficacy of the neutralizing antibodies, again, this is before the challenge. What we can see here, that there is a nice binding antibodies that are developed in the sera of these hamsters, as you can see here in this stain of vero cells infected with the SARS CoV-2. And this is the data regarding the neutralizing antibodies, again, 3 weeks after the vaccination. These are the different doses, and you can see nicely the dose response. We have to mention that this low number, as you can see here, is at the range of convalescent people. So even these numbers should be enough in order to protect. regarding the second part of this experiment, after the challenge, you can see nicely at this again weight change graph. In black, these are the infected and treated animals, and all the colors represent the different background of the vaccination. In all of these cases, there was a significantly protection regarding the weight loss. In addition, all of these animals, these vaccinated animals, when we examined their lungs for the virus, all of these lungs were clear from the virus compared to the infected and treated animals that there were a high number of viral particles in the lungs 5 days post-infection. We also examined in the histology of these lungs 5 days post-infection. So on the left side, this is the naive sample, the vaccinated in the middle, and on the right side, you can see the unvaccinated, infected, untreated sample lung, and this is a quantitation of the tissue to air space that is very high in the infected compared to the vaccinated hamsters. And this is the data regarding the particles, the viral particles in the infected compared to the vaccinated hamsters. The second model is the model of the rabbit. This is the medium-sized animal that we are used in order to evaluate mainly the safety and the efficacy of the vaccine. In this case, the efficacy is being evaluated only by the immune response, the binding antibodies and the neutralizing antibodies. Again, this is the schedule of the treatment. And regarding the safety data, again, this is a prime boost regime. This is the body weight of all the animals, vaccinated and unvaccinated. As you can see, there is no effect regarding the body weight or the body temperature. And this is the data regarding the neutralizing antibodies. This is the low dose of 10 to the 6 that was examined in this experiment. Following the prime and the boost, you can see nicely the effect of the boost in all of these vaccinated rabbits, 10 to the 6, 10 to the 7th, or 10 to the 8th. So this is there in the rabbit, there is a nice effect of the boost regarding the immune response. And the last model is for our case, is the big animal model, is the pig model. And again, there is a lot of data that we generated regarding the safety following the prime boost vaccination. And again, the efficacy is being evaluated in this case regarding the immune response. In this case, we also added the T-cell response. And this is again, the schedule, the tracking parameters, a lot of tracking parameters that I'm showing only part of them. Again, following the prime boost regime, the body weight of the pigs that were vaccinated compared to the control and the body temperature, all were safe. And the neutralizing antibodies, again, nice induction of neutralizing antibodies following the boost, both in the 10 to the 6 and to the 8 of the 10 to the 8 vaccination. And this is the data regarding the T-cell response. Again, this is a ELISpot assay for the T-cells. And what we are showing here is the induction of the T-cell response following the boost of these pigs. Transfer it back to Nir.
So the last part is about the generation of monoclonal antibodies, therapeutic monoclonal antibodies. So the aim is to isolate a panel of human monoclonal antibodies that will serve as the basis of efficient post-exposure therapy for SARS CoV-2 infection. So we wish to isolate antibodies, and we isolated antibodies to the spike structures on the surface of the coronavirus, and especially to the receptor binding domain, the RBD, which mediates the interaction between the virus and the target cells, and this preventing this interaction would prevent the infection and the development of disease. This work was also submitted for publication and was accepted for publication in Nature Communication. So we try to isolate antibodies from convalescent people as well as from coronavirus infected or vaccinated animals. So from all these samples, we generate combinatorial phage display libraries with the idea to select antibodies that bind specifically to the receptor binding domain on the spike protein. And then we generate a repertoire of antibodies by cloning and characterization that and we wish to have a cocktail of antibodies in order to improve the neutralization efficacy and to minimize the chance of escape mutants. And having these antibodies, we wish to have a therapeutic product to treat severe ill people. So we currently have a panel of 8 human neutralizing antibodies that were generated, and these antibodies are have variable sequences, and we showed that all of them specifically bind the spike protein at the RBD. This is in gray. The S1 region of the spike, which is important. Both are important for virus entry and, of course, for neutralization. They don't bind the N-terminal domain of the spike, which is here in black, which is important because this region is not important for neutralization. And so all the antibodies are related to actually where we map their function and their binding activity, and we show that they are related to 4 different families, and they bind distinct epitope, and we are mapping these epitopes on the spike protein. There are antibodies that have strong mutual binding, partial or no mutual binding, and having the repertoire of various antibodies would allow to have both high neutralization titer and to prevent escape mutants. So here is a demonstration of the virus SARS CoV-2 neutralization activity of the virus antibodies, color-coded by their family. And you can see, for example, in green, antibody number 65, which is a very potent neutralizing antibody, which is from the second family, the green family. From the pink family, number 62, which is also very potent. So there are various antibodies with varying neutralizing activities, and we are characterizing them all to have the best cocktail we can have. And we wish to have a cocktail that will altogether will prevent infection with SARS CoV-2. The current status is we are doing both preclinical studies, meaning to have data on the efficacy, the pharmacokinetics, the toxicity of the antibodies. We are broadening the antibodies repertoire by having another set of human antibodies and isolating antibodies from animal-derived sera. And to summarize both subjects, so we presented our work on the generation of PVSV-AG-spike vaccine and monoclonal antibodies. We showed that the vaccine is potent and safe, and we are currently at the preclinical stage, and we wish to continue towards the clinical studies. And with the monoclonal antibodies, we have a repertoire of antibodies binding various areas or epitopes on the spike protein, and we are currently addressing preclinical issues towards production and clinical studies as well. So there are many people in our institute that contributed to this work. It's a very demanding work, which includes a lot, which most of the work is done also in the BSL-3 lab, and we would like to acknowledge their contribution and to acknowledge also the financial support from the government and the supply of clinical samples from the clinical centers. So, and thank you very much. And we would like also to thank the organizers and Professor Schenker for inviting us to give this webinar. Thank you very much, and we are all open for questions.
Thank you.
Thank you very much for your scientific contribution, and we are looking everybody around the world for his practical application. So now please send the questions, and if anybody has questions or send questions. Okay, we have some question. It's from Walter Feller, and he is asking, what is your opinion about the horses' antibodies serum to provide passive immunization that under development in Argentina?
So concerning horse antibodies, there are two issues. Of course, they can be efficacious, and in horse you can do large-scale production of antibodies. The problem might be that if you inject horse IgG, even only the FAB part of the antibodies, you can only inject up to, I think, 3 times, no more. And so it will prevent people from receiving other horse products, and this might be a problem. Also, there is a major issue of antibody-dependent enhancement of infection. So you have to make sure that you have very potent neutralizing antibodies, and having the ability to generate human monoclonal antibodies, we believe that that would be a more dedicated and efficacious product. But we have to look to see the data from the horse product.
Regarding the question of Smita Lula, what are the side effects of the vaccine? So up till now, in all of our preclinical studies, again, in hamsters, in rabbits, in pigs, we don't see any side effects. So we hope that the side effects also in the clinical study, in the phase 1, will be minimal. Again, with all these animal models, we don't see for now any side effects.
I would also like to comment that having the data from the Ebola VSV vaccine that was given to more than 300,000 people in Africa, we believe, and was approved by the FDA and the European regulator, we believe that the vaccine would be well tolerated in the population. Concerning the question of the dose depending, to changing the dose of the vaccine depending on the weight of the animal, so it's a very good question. It is considered for vaccines that when you address their efficacy or safety, you inject them in the dose that is the human dose. So even if it's a hamster or it's a pig, you inject the human dose and you evaluate the efficacy, because what is more important is the interaction between the vaccine and the immune response of the animal than the weight. The weight is important concerning the safety, and then if we vaccinate with a high dose, it would better simulate issues of safety concerns if there are. And as Tomer said, we don't yet see any safety problems, and we are really trying to find them to make sure that the vaccine is safe.
Regarding the question of Nizlot Mittler, if I got it right, the biggest model that we examined is indeed a pig model. And actually, we are now started the last animal experiment, and we hope that it will go smoothly. And if indeed we are planning to start the phase 1 clinical studies around the late September, this is the plan for now.
So I have additional question. When you think the passive vaccine will be approved?
It's a very good question. We are working now to gather or to collect all the preclinical data that we have, together with production issues, and we hope that in few months, I hope, we will have a better answer towards this question. The question is, of course, we have in Israel the Israeli Ministry of Health, which is the regulator here. The question is whether we apply for the Israeli regulator or for regulators abroad. And we are dealing with this issue now to have the fastest way to approve the product.
Anybody has additional questions?
So there is a question about vaccination of autoimmune disease. There is, of course, you start the clinical studies with healthy individuals, and you exclude people with immune problems. And then after approving the safety in healthy individuals, you start to challenge the system with people with immune problems. I don't have any data concerning contraindication to autoimmune diseases and VSV, but we will have to follow the first phase 1 and phase 2 before we will start to deal with people with autoimmune diseases.
So following your last answers, the question, my question is COVID more infection disease or immunological condition?
It's a very tough and important question. We see, of course, there is the etiological agent, which is the SARS CoV-2, and what is the role of the immune system, the innate and the adaptive, is currently investigated. We are also asking this question in our, or there is a major debate about the role of cytokine and cytokine storm in the disease, but as far as we see, at least concerning the immune response, if you have an active and healthy immune system, you are you will have a good chance to overcome the infection. But what is the reason why some people develop a very severe disease is currently not answered fully.
Thank you very much. Additional questions, maybe?
Another question by Lance Webb regarding, do you have an opinion regarding whether the vaccine or the monoclonal antibodies will provide the longest lasting immunity? So historically, basically the vaccine by itself should provide much longer immunity. This is the reason why it is aimed to the healthy population in order to get a long-lasting protection. The antibodies, the monoclonal antibodies are made to be a short treatment to sick people. They will not last for long, they will be clear. So this is the main differences between the vaccine and the monoclonal antibodies.
Yet, this is considered a very efficacious vaccine, and even after single immunization, we expect to have long-lasting immunity. The data so far, even from Africa, is that people have immunity for several years. So we expect this vaccine to be efficacious as well.
So, on the behalf of the Academy and our board, I would like to thank first of all to our distinguished presenters, Dr. Tomer Israeli and Dr. Nir Paran, for their excellent presentation and for their insight in the work behind the science to develop COVID vaccine and giving us hope, which everybody around the globe is looking. I would also like to thank the Biotechnology Congress for organizing this webinar and for assisting us with the technology problem, especially thanks to Mrs. Valentina Tonelli and to David Genezani for your support. Before ending today's session, it is my pleasure to invite you all to join our 19th World Congress of the Academy of Human Reproduction that we are looking to hold it in 2021 in Jerusalem, and it seems that it much depends on your success. Thank you to all participants, and I hope to meet you again in our next meeting.
Thank you very much.
Thank you very much.